I've been going for a long time now, trying to go conventional. And by conventional, I mean we got to get past a bunch of this stuff that we do, whether it's psi or whether it's healing or whether it's whatever it may be, and make it so it's not threatening. And make it the way to make it so it's not threatening, I think, is that we have to make, in the case of healing, we have to make healing not, this wild-eyed, oh wow, that's the most amazing thing that's ever happened. My pain went away, my condition went away. I mean, enough already. The question is, how do we get this out there to the world? And we get it out there to the world in large numbers so that it becomes nothing that's exotic. So I'm going to take that route and we'll see where we go with that. The plot that I want to talk about here today, briefly, is to contrast the regular history of healing with changing the focus to storing and scaling. And I think once you see that what we can do with storing and scaling, a whole new vista is going to work and open up for you. So a couple of quick things on healing. So let's go to the conventional stuff. And when you think about healing, you think about where my mouse went. It's kind of ironic when I lose mice. So if you think about healing, I went too far. You think in something like this. You think of a healer and you think of a healee. And you also have a tendency to see something that's going on here. Everybody's quiet and somber and it looks like it's a sacred act of some sort. And this guy's got his eyes closed and he's probably zoned out in something and she's doing whatever she's doing and I'm losing my place in line. Let's try it again. So we got a healer and a healee, and I spent about 50 years looking at this. What happens between healer and healee? And I got a lot of stuff on that. And a lot of people have looked at a lot of stuff. What happens to the healer? What happens to the healee? I'm not the only one, obviously. And what I want to do is take whatever she's doing, stimulated by him, who's the healee, and put it in a box. So that anybody can go to the box when they want and take out a little healing. You get, I need 2 cc's of healing, whatever it might be. So how do we do that? Well, we're used to storing stuff already. So if we take electricity and it's exotic and you come back a couple hundred years, it's this fanciful thing. But by today, we've put it in a box. And if I say we've got electricity in a box, you don't start flopping on the floor and getting all sacred on me. You say, yeah, it's a battery. So once we can label it a battery, as opposed to healing, we've gone someplace. And so if we can store it, we might also be able to scale it. So wouldn't it be neat to make a battery and make a bunch of batteries so everybody could have a bunch of batteries? Well, it turns out we can. Can we do the same thing with what was once? We look back on 2023 and we go, remember when people thought this healing stuff was all this sacred stuff? It's just sticking electricity in a battery. It's just making food stuff. So you need some fish oil, I can get you fish oil. I can get you fish oil in large quantities. And I can store it in little fish pieces. That's pretty interesting. And that's what traditional, before you get to the storing and scaling, you got a healer doing the healing stuff, and you got a healee doing the healee stuff, and we try to figure out what's going on. So let's look at some previous research on healing, and this is old stuff. This is stuff that's been verified. It's enough already, and we don't need to keep revisiting and revisiting and revisiting. So I can show, but I'm not going to because it's not the point of the talk. I can show reliable empirical data, such as if we're looking at healing, there's cures all over the place. But look, I'm not going to go through this whole thing. I'd invite you to stop and think about it for a bit. The second bullet, well, it's the only one I'll talk about. The second bullet, distance is irrelevant. Now, how in heaven's name could distance be irrelevant if it's energy? So I got a patient who's in Australia, and I can either go to Australia and do hocus pocus on the patient, or I can do it from here. What's the difference in outcome? There isn't one. Now, if there isn't an outcome that distance matters, and we spend a couple of minutes thinking about this, we already know that it's not energy. Energy dissipates with distance. All energies do. And it's a pretty simple formula. But in healing, there's no dissipation. It doesn't seem to make any difference. And we've done this with cell cultures, we've done this with mice, we've done this with people, we've done this all over the place. This is not a big exotic thing I'm saying here. Distance is irrelevant. If I put a mouse in California and I treat from wherever I am, the mice get better the same way as if I was in the lab. Let's spend a little time thinking about the implications, but we have reliable empirical data on all of this kind of stuff. And we could spend the rest of the day talking about all these bullets, but we're not going to. It's not the main focus. So we know some stuff. We don't know most stuff. And so I just have, we don't know, oi. So it's just a oi. Because this in particular, we need the second slide, the third slide, the fourth slide, because the reality is we don't know too much. And that's the reality. So look what we don't know. First bullet, is healing energy? Is it information? How does it work? I can't help but I got to do the second bullet. How do distant treatments hit a target? So I got a mouse. I got a mouse in California. It's 2,500 miles away. And I do hocus pocus as if the mouse was in the room, and the mouse gets cured of stuff. How in heaven's name did it find that mouse? I mean, come on. When someone comes along and says, I know how it works, and they'll say something like quantum this or quantum that, please. Nobody knows. Why don't we just say we don't know? So how did distant treatment hit a target? I don't know. I don't know. I don't know. I don't know. I got a whole lot of I don't knows up here. I think. I don't know. So we contrast traditional kinds of healing studies, and now we look at storage studies. And I'm just going to tell you in nice simple terms, I've done a lot of storage experiments. Can I put electricity in a battery? Can I put healing in a battery? Yep. Can I put healing in a, yep. Can I put yep. And it's really interesting, but not the point of this talk. So we've tried storing in a bunch of stuff, organic and inorganic materials. So for example, we've got a bunch of experiments at Brown University on cotton. So here's exhibit A, or my show and tell. We take cotton and we put cotton and we hold the cotton and we do our healing stick. We don't get sacred and flop on the floor. We just do our healing stick. And then we see if the cotton's different. Well, I've put cotton under an electron microscope. It's not different. It's not structurally changing anything. But if I put cancer cells in vitro, so I think I'm in a little rectangle up there. If I put cancer cells in vitro and the cotton has been charged, as the cotton approaches the cancer cells, they genomically change. In other words, something is recognized. That's reasonably interesting. If we take cell medium, stuff that we use for in vitro studies in a lab, we take the cell, the nourishment, and we treat the nourishment, and then we put cells into the medium, we get wild results. In other words, something's happening to the cotton. It's not structural. I can't look at it and see its little quantum things going on. But what I can tell you is that cancer cells or cells in need can recognize whether cotton has been charged. That's reasonably interesting. We can treat mice and take blood from that mouse and put it into an infected mouse. It'll heal that infected mouse. That's interesting. And what it's pointing to all in one direction is pointing in the direction of, yes, it can be stored. It can be stored all over the place. We can store it inorganically. Water, media. So water itself, here I got another exhibit, exhibition B. Water in and of itself can be treated, and the treated water fed to mice, and the treated water fed to cells, and the treated water applied, and it's as if you were doing it with hands-on. Now that's reasonably interesting. So we have something we call treated water. And I'm going to contrast that in a little bit. Treated water is water that has been held and you have intention of hocus pocus. Informed water is what I'm going to contrast that with. Treated water takes your healing technique and puts that healing technique, we think, using it loosely, into that water. Informed water is a term we made up, and informed water means water that has been mechanically scaled by a device. So that if I have a bunch of treated water, I've treated each piece of water, each vat, each cylinder, each container. If I have informed water, I've stuck it in a gizmo and I've tried to reproduce this without any healer. And I'm trying to get to the point where I'm doing this stuff without any healer. So if we record healing, it's inorganic. If we record healing and we play back the healing intention on a recording, you can think of the scalability potential here. So let's look at some stuff. And this is only a small selection of stuff. This matters, this slide. So we've got on the x-axis, how scalable is the thing? And on the y-axis, how good is the thing? So if we have scalability low and effectiveness high, and I got a little dot there, that means if I do hands-on treatment, it's very good. Hands-on treatment works. But hands-on treatment is limited to the hands-on of what you got in front of you. So like in the picture, I have a healer and I have a healee. Will that work? I mean, we're doing a hypothetical. Will it work? Let's assume it'll work. That doesn't mean we can scale it. It just means we've shown healing. And so it's very low on scalability and very high on effectiveness. Now what we're looking for, of course, is effective and scalable. So we have over here cotton and treated water. If I do, and this dot should be closer to this dot. If I take treated water, if I take water and I treat it, and we give it to cancerous mice, the mice get cured. That's research I did at University of Connecticut Medical School. If I take cotton and treat it, we've already seen something happens. It's very effective. But what do you do with cotton after you've used it? I don't know. I mean, how do you make a whole bunch of cotton? I don't know. What if we had, instead of a healee, we had 100 healees? Hands-on doesn't translate well to 100 healees or 1,000 healees or 1,000,000 healees. And there's at least 1,000,000 healees who need help. We're not going to get to them one-on-one, putting your hands on the healee. We're not going to get to them one-on-one saying, here, take a piece of cotton. We're not going to get to them one-on-one saying, here, take some water. Does it all work? Yes. Does it scale? Not so much. And I contrast that, if I can find my marker here, I contrast that on scalability high and effectiveness high. And I'm calling this, or I'm categorizing some recent studies that we're calling informed water. So once again, informed water has no human involved. The thing can be automated, and we're creating something that stores, we're putting it into water, and we're not sitting there putting your hands on somebody, or we're not giving them a piece of cotton, or we're not saying, drink this water. We're closer to drink this water, but we didn't make the water. So we're not walking around for an hour holding a glass of water and saying, here's your therapy. We want something to come out of a spigot, essentially, with unlimited amounts coming out of the spigot, and we're calling that informed water. Treated water is water that's been treated one-on-one, and so is not particularly, it's effective, but it's not particularly scalable. Informed water is water that is scalable because we can make a boatload of it, and it's also effective. And so up here in the right-hand column, I can't find my pointer. Right here in the right-hand column, that's the sweet spot where you want to be. You want informed water. You want something that can be reproduced at will with no limit to the amount you can reproduce. So for example, a recording. If we can make a recording of healing and reproduce the healing by playing back that recording, we have unlimited scalability. So in essence, the short version, you store it in the cloud and you say, you want a treatment? Download this file. That would be maximally scalable And you can see how we could do 1 billion doses without too much trouble. 1 billion people could listen to the recording and produce healing all over the place. If the water's informed and it works and it's scalable, we could take 1 billion people who need water, give them the water, 1 billion of them, and say, go off into the sunset. Informed water, by this scale, is as essentially scalable. Oh, it's not really, but it's essentially as scalable as the recording. But what we found, and I'll just drop this bomb, the recording works, and you'll see it in a second. The recording works, but it doesn't seem to be as strong as informed water. Meaning, mechanically produced water seems to be stronger, even if it's slightly less scalable, as does a recording. Recording has the advantage of instantly globally scalable. The informed water has the characteristic of, it does a great job. In fact, the informed water we're finding, at least by anecdote, will do stuff that the hands-on won't do. On the flip side, we found that the recording doesn't do all the stuff that the hands-on can. So our sweet spot is over here. Our ultimate sweet spot is the informed water because it seems to be effective and scalable. But the recording seems to be scalable, obviously, but not quite as effective. That doesn't mean we can't make it better, because we can demonstrate that this is a technology, and a technology can be futzed with until it's made better, stronger, etc. So let's stay in the top right corner here, and we'll leave the hands-on healing goodbye, the cotton goodbye, the treated water goodbye, because it doesn't have enough scalability properties, but we can play with informed water, we can play with recordings. So my research agenda, and that's really what I'm talking about here, that I can't get out of, my research agenda has 2 arms. Since we have success already with a recording, and we have success already with water, both treated and informed, but particularly informed, we want a whole crew working on the recording and continue to work on the recording. If it's a technology, we can make it better. Same thing with the water. We can make water. We know water works. We know informed water works. We know we can scale it. Can we make it better though? So that's what we're working on, making the recording better and the water better. So let's look at both arms real quickly. So first we go to the recording for maximum scalability, but not quite as much. I'm breaking this down into basic research and clinical research. And as you can see, I have more stuff on basic research and the recording than I do clinical stuff. We don't have much clinical stuff. If the stuff is in red here, it means we haven't done it yet. So we have a bunch of stuff using the recording on cells and mice, in vitro and in vivo. This summer, winter, next year, sometime in the next year, we're hoping to get a bunch of clinical studies done on the recording itself. So let's look at the recording itself and stay over there for a little bit. In the recording, as some of you already know, we take a big Faraday cage. This is actually Dean Radin's Faraday cage in the old ions. And we put in this, since we're not exactly sure what we're doing, a whole bunch of detectors. Just put a whole bunch of stuff in there. And 3 people who are familiar with my healing technique, one being me, takes a piece of cotton and goes in the Faraday cage. We take 3 people in, and we run the 38 detectors, and we have them all charging a piece of cotton, not for the cotton's sake, but to get the treatment going. And then we have a recording reduced to a WAV file of what just happened in the Faraday cage, short version. We then take that recording and we play it, ordinary amplifier, ordinary speakers, ordinary incubator, we play it to cancer cells. So what happens if the cancer cells don't come near a person, but rather they're just exposed to the recording? And it turns out lots of stuff happens. I mean, a lot of stuff happens. So this was particularly interesting because for the first time we got this in a biology journal, a traditional biology journal, and basically we looked at what happens when cancer cells in vitro are exposed through a recording. And it turns out lots of interesting stuff happens. Short version, we looked at 167 cancer genes and we found 68 of them genomically change reliably. This is done at Brown University, 7 replications. We can demonstrate now healing can be captured and released. So the potential is there. But we still find, I'm on number 3, we still find hands-on healing is stronger than the recording, at least the one we have. So among the research agendas that I have is how do we make the recording stronger? How do we make it so it does all the things that hands-on will do and is instantly globally disseminated? That would be interesting. And perhaps the most important finding is number 4, which we actually didn't put in the biology journal because we thought we'd scare people. It turns out when you play the recording in a room, the room itself changes. And the room, without the recording lasting there or leaving it there, the room will hold the healing intention even without the recording, and it'll hold it for weeks. So we can change, short version, we can change cancer cells genomically by putting them in with a recording, and a bunch of the cancer cells will flip. But we can also turn off the recording and put the cancer cells in even weeks later and leave them in for a few days, and the cancer cells still change. Now you spend a little time thinking about that and you're going to get a headache. And the other thing that's going to happen is you're going to say, you don't want to tell people about that. If we have conditioned space that lingers in a room, we're misinterpreting a whole lot of experiments that we're now doing. Anyway, something to think about. Anyway, so we took the recording and we did some experiments now in vivo on mice. This is also at Brown. And same idea, we got mice cages. We don't care about the mice themselves this time. We got a couple of speakers, we got an amplifier, we got a computer, and everything is automated. And we play this recording to the mice, and what happens? Well, we start to get biological changes, which we got with hands-on. It's just not quite as strong. And we get the ulcerations many of you are familiar with, and we get the thing going towards a cure. But the problem we had was not the mice, was not the recording, but rather it was the ethical committee at Brown. And the ethical committee at Brown, they're called an IACUC committee. They came along, looked at them at one point and said, well, that's really ugly looking. They must be suffering. And we argued they're not suffering, they're just hanging out and they got ugly things sticking out of their bodies. And they killed them. So we had an aborted study. I'm not sure why killing them when they weren't in pain to begin with is an ethical thing to do, but who am I? We salvaged enough data to make another biology journal successful publication, and this time looking at mice. And some of the stuff was pretty interesting, what happened with mice and the recording, but I want to move on to water. Look at the last dot. Spleen weight increased 5-fold in these mice. Now that's not a small amount. 5 times the spleen, that tends to be a marker of immunological responses. So without going into this in any detail, we found some pretty serious effects. And you can read the paper if you want. You can get the paper off my website. I just recently finished a couple of studies at Tokyo University doing in vivo experiments on mice, and we had different kinds of cancers. We had breast cancer, bladder cancer, melanoma cells in mice, and we did the same thing as we did at Brown with the breast cancer. And it turns out that the breast cancer had the strongest effect, and had the strongest effects in volume and mass, and a bunch of blood measures that we don't really care that much about here. And an interesting thing that we found was that it didn't affect melanoma cells. So the recording played to breast cancer cells. The breast cancer cells know it's there and do stuff. Melanoma, no. Melanoma didn't seem to be significantly affected by the recording. And we left with a big question mark about bladder cancer cells, what I'm going to duck for today. But this too, now we're 3 for 3 in biology journals. So biologists are loosening up a little bit. And we're looking at really exotic stuff, really unorthodox stuff. And it's unorthodox stuff, I'm not sure how many people pick this up, can be scaled because they're listening to a recording. So short summary, recordings work. They don't seem to work as well right now as hands-on. They don't seem to work as well as the storage in water. They don't seem to work as the mechanical storage in water, but it's interesting and it's worth working on. So I need a team, and I'm looking for a team to keep doing some further work on the recording because we're yielding some interesting data on that. The second arm of my research is with water. And water comes in two flavors. It's not really a flavor. Treated and informed. Treated water means I'm taking water and I'm doing my healing shtick to it, and then you're applying that to mice, to cell cultures, to whatever you're applying it to. And we've done a bunch of those experiments. Informed water tries to replicate that with no human intervention and everything being automated. So the informed is obviously more scalably interesting than the treated, because the simple fact is I can't treat that much water, but I can have an output of it for informed water that hasn't seen a human. It's rather been automated by a machine. So here on the water side of this, we've looked at treated and informed. Informed is more interesting. And we've done basic research. Since it's in black, it's in vivo, it's been done on mice. Clinical, we got 3 human trials of informed water and a COVID trial on informed water. And we have a bunch of things in the pipeline to be done. So the to be done includes long COVID, which I'll get to in a second, which could turn out to be a reasonably interesting thing to look at. So informed water is stuff that no human has treated. It has come out of a machine that stems from treated water, now converted to informed water. And so informed water is in these 2 bottles. It's supposed to mean 2 different kinds of informed water. And we put the 2 informed waters, depending on what we do, we give it to a clinical case, we give it to a in vivo case of a mouse. We put it, but it's coming out of a bottle. So it makes no difference who applies this. And we do stuff on double blind, and we do stuff on single blind, and we've done a whole bunch of stuff. In single blind stuff, we've done 3 clinical studies with energized water of some sort. And the thing that we really care about is the informed water. We've done 2 100-person clinical trials in the States, and we've done 100-person clinical trial in Europe, and we get exactly the same thing. And the thing that we get is remarkable findings of people who have biological need, emotional need, spiritual need. They're depressed, they're flopping on the floor, they got bipolar, they're whatever, they got. And we ask them to track their conditions at baseline, and then every 2 weeks for 8 weeks. And then we followed, tracked, and did numbers on how are their conditions changing, how is their life changing. And the stuff we got is frankly a little over the top. It makes my head hurt. We got 98% of the 300 people saying that in some facet of their life has significantly improved, and virtually everybody wants more water. Now that's interesting. I don't know what to do with it though. So we have people who are on bipolar meds, they give up their meds. We have people on depression meds, they give up their, we're not asking them to do this. They're just, they transition somehow. They got tumors that fall off. They got. Now it doesn't mean 98% of the people are cured. It means 98% of the people have had some thing that they consider significant to their lives, and they want more. So this is a single blind test. We also did a really tightly controlled double blind test, which we just completed not too long ago, on 160 people on their way into the hospital to be treated for a condition they know they have. So they've come in and they've taken the COVID test, they've come out positive, they got the symptoms, they're hacking, they're flopping on the floor, they need to go to the hospital. We catch them on the way into the hospital with their COVID certificate, and double blind, and this was seriously blinded, double blind, randomized, placebo controlled. We had doctors giving everybody water. They had no idea what was in the water. So the doctors were giving, with instructions, we never met the doctors in the hospital. The doctors in the hospital were administering sublingual drops from these containers, 2 drops under the tongue, 6 times a day. The patients didn't know what they were getting. The doctors and nurses didn't know what they were getting, and everything was independently verified. All the water came as informed water, meaning no human ever played with it. And we published this in JSE a couple of editions ago. I don't know how many, a couple of months anyway, a couple of months ago, a double-blind placebo-controlled clinical trial on hospital COVID patients using informed water. Now, the real short version, and you can read the whole thing, obviously, in JSE, the real short version is this: blinded, placebo-controlled clinical trial, enormous effect. The control group was not somebody who took no medicine. The control group was somebody taking medical intervention. In other words, they went into the hospital, they were told to take this medicine, they took the medicine, and they went along their merry way. They're still in the hospital. And we monitored their fevers, their coughs, their this, their that, a bunch of measurements. And it turns out, compared to people who just took medical care, short version, you can see the details, read the paper. The short version is the informed drops of water, which means water. If you chemically look at it, it's water. The informed drops of just water blew away the medical treatment. If you have a serious condition, let's say you have a very bad persistent fever, well, this will take care of the fever very fast. Medical stuff, if you're just in the hospital and you're getting a placebo, eventually get better. These weren't people dying left and right, but they were seriously ill. But comparing what happened to them with placebos and what happened to them with the informed water, we blew out the medical system. I don't think there is a stronger effect size in COVID than what we got. So it's reasonably interesting. And we can expand this a little bit and look at the right side, water, treated and informed, but more interesting is informed. We've done basic research on treated and informed. We've done clinical work, treated and informed. We've done research on mice, treated. We've done human trials using informed water. We've done a COVID double-blind placebo-controlled using informed water. And we've got a couple things going on. We've got a study on worms going on in Brown University right now, because we want to see if the water has any effect on telomere lengths or lifespan or things along those lines. So we're using C. elegans worms, which is the most commonly used model to check stuff like this. That's going on right now. I might have the response to that when we get to the Q&A in a couple of weeks. I might be able to report on the worms. It just disappeared. Okay. On the water, we've got clinical trials, human trials, COVID, and we're about to start a couple hundred people on hepatitis B. And this is being done in a country that doesn't have access to a bunch of hepatitis B vaccines. So we're going to see if we can take care of hepatitis B. We're going to take on I'm not sure where or how, but we want to take on long COVID. So COVID isn't as fatal as it used to be. Long COVID is pretty annoying socially, and we're going to see if our drops, again, no humans touching them, if our drops attack, fix, or something along those lines, long COVID. And I also want to try, and I may be able to start this in a particular country, an antibacterial model. We have an antivirus. The antivirus gets rid of COVID, it gets rid of the flu, it gets rid of Ebola, I mean, we've done a lot of studies that I'm not mentioning here. It gets rid of a lot of things viral based, but we want to see if we can attack things that are bacteria based. And bacteria based are usually done by antibiotics. And antibiotics are starting to lose their luster as they become weakened with overuse. So we want to see if we can turn water into the equivalent of an antibiotic. That would change potentially a lot of stuff. So hepatitis B, long COVID antibacterial model, worms, intention coupled with the recording, a bunch of stuff it's in there. And no matter where you look on this, since recording and water both work, we've got a bunch of potential to look for storability and scalability. And we've got a bunch of potential to look at that, whether it's a recording, and I have more data on different types of recordings and also different types of water. So I could keep going, but I'm out of allotted time, I think. And I'd be happy to answer any questions or correspond or put you in the direction of a paper that might answer your question.